Already gone over molecules and the difference between the first part of the class was on atoms and individual elements and the difference between those, and then the second part was that we went over was molecules. Molecules are a more complex structure that's made up of multiple kinds of atoms, so not a solid element, but like for the example here, water, where you have an oxygen atom and two hydrogen atoms, and that's a molecule. So that was what we covered in the second part. And then what we're going to talk about now are organic compounds. And so these are more complex structures, and in a lot of ways they're repeating structures. So in all organic compounds, except for there's some very, very weird ones that use sulfur instead of carbon, pretty much everything that we call an organic compound on the planet is going to have a carbon as part of its base. And in some cases, they're written like this, where they actually include the carbon in the diagram. In other cases, they're written in this method. You can assume that on organic structures, when you see organic drawings, any one of these corners is going to be a carbon. So this is a benzene ring, and you have carbons at each one of these corners. So, and for example, this is glucose. So when you see these drawings, if you see something here that has, a carbon group at one end and then just like a squiggly line going off of it, each time that little squiggle goes, it's going to be a carbon and it's going to be two hydrogens, unless there's something else drawn on there. So like in this case, you have a carbon, a hydrogen, and a reactive chain that's going to go off of here. You have a nitrogen and two hydrogens over here. But for organic compounds, and that's what this is going to cover, they're all going to have a carbon chain in them or a carbon base. What we're going to talk about is, we're going to start off by talking about monomers. And monomers are small pieces. And you take those monomers. So monomer, it means single or one. So you have small organic compounds. You put these into chains and you get polymers. So, and if you think about it like this says, you think about it as a single link in a chain is a monomer. The entire chain is a polymer. All right. And so you can see that here just represented in this fashion. And once again, so these circles here now are another form of representing different atoms. In this case, these circles would be your carbons, and the small dots would be your hydrogens. So you have these monomers here, which are small individual pieces, or you have a polymer, which is a repeating chain. So, and these are your larger polymers are called macromolecules, and that's when you get stacked up polymers combined together. So here's an example of the polymers and monomers that you're going to find in 4 different forms in biology. So, and these all should seem relatively familiar. So the first 3, I'm going to kind of go a little bit out of order here, but the first 3 are proteins, carbohydrates, and lipids. So proteins, sugars, and fats. And they're all structurally a little bit different. And then the 4th one is your nucleic acids. So those are your DNA and your RNAs. So obviously, all of these are important for the building blocks of life. So we're going to kind of go over all of these and what their monomer is for it. And then also, as you start to go up to higher and higher levels, what your follow-up pieces are. So this is just throughout this PowerPoint, I'm going to have a few little questions here. So I'll pause and I'll let you guys chime in. You don't have to raise your hand or anything, just first person to speak. On this list of these four, which one is the organic compound? And you can just say A, B, C, or D.
C.
C. Okay, and why?
Because it has carbon.
All right, exactly. So all of these are molecules. This one's an organic molecule. All right, and it's a C6H12O2. All right, so we're going to go into carbohydrates. And carbohydrates are essentially you're starting your monomers for carbohydrates are going to be your sugars, right? And your sugars are very important, and carbohydrates are very important. They're basically our basic block for the conversion of from sunlight into usable energy by plants and animals and pretty much everything else. So it's through photosynthesis you take sunlight and you're making sugars. So let's look at what sugars look like, and then how, as you start to make these sugars into more and more complex features, how you get then into starches and into carbohydrates. All right. So we have our monosaccharides, right? So these are single sugar rings. And in this case, we have a glucose and a fructose. You can see that they have different number of carbons in here. And like I said, that you can think about this like a zigzaggy chain, but these are rings. In the rings, each one of these bends is then going to be a carbon. If they're simply drawn like this, they don't have anything else added onto them. You just assume that coming off of that carbon is a hydrogen, or in some cases two hydrogens. So you'll have a hydrogen bond and a hydrogen bond. They don't draw those in. So your glucose is 6 carbons, your fructose is 5. Then you have your disaccharides. And so for they're a 2 carbon ring. And that one's this is just a very common sucrose, is a glucose plus 1 fructose. And then also here's just 2 glucoses combined, and you can see that they do bind in different places, but it's always that bond between your carbons. And we'll talk a little bit about how these bonds are made through dehydration synthesis and through hydrolysis is how you break them. And basically, when this bond is formed, a water will be left over. And when you break this bond, you're actually adding a water in. So you'll get a hydrogen will be over here, and an OH group will be over here. So we'll look at that in when this is all done. This first slideshow is mainly looking at just kind of some of the basics. All right.
Does
All right.
it matter?
What's
Sorry.
that?
Does it matter how many sides it has?
It does change how it works. So like in like your Krebs cycle and things like that, whether or not you're bringing in a fructose or a glucose, yes, definitely matters. And it also changes like the organic structure, and so you get like different organic structures will bind together differently. They kind of bump into each other, so you get what's called steric strain. So these are your polysaccharides, and your polysaccharides are they're chains of those monosaccharides, right? So and a few examples of those. And so as we're moving up, right, we're getting more and more complicated. So we had our monosaccharides, right, which are singular, our disaccharides, which are 2, and then our polysaccharides, which are chains. And the bigger your chain gets with your carbohydrates, the more energy that's stored in these. So this also, because there's more energy stored in them, it takes a longer process to break these down too. And so you hear about that as far as like glucose and fructose being just very readily available energy. And part of that is because it doesn't take a lot of effort to turn these back into ATP, which is basically the energy process, or the energy currency of your mitochondria. So it's what we use to then do other processes within our cells. So like opening up gates in the phospholipid bilayers of the cells, and transporting things across those layers, and all of these other things that take energy. Plants turn sunshine into these complex shapes, and then through the process of the Krebs cycle and Calvin cycle, we then break those complex shapes back down into their bases, and by doing that, pull energy out in the form of ATP, and then that ATP can get used to build other things in our body, and also to do processes that are in our body. So if you just think about it, they a lot of times they just talk about it as energy currency, which I think is a really good definition of it. It's essentially like money. So and so they're the sunshine's coming in, it's getting stored in these, and then they're releasing that energy later on in the process. So like I said, we go from monosaccharides to disaccharides to polysaccharides. And all of these names should be relatively familiar, right? Obviously fructose and glucose, and sucrose for table sugar, and maltose for it's your beer sugar, right? And then you get your polysaccharides. So you have starches, which you have in a lot of plants. And it's a reason why, so like a potato, there's a reason why a plant stores its energy in starch, because it's a big long chain that's full of a ton of energy. So for the next year's growth of that plant, essentially they build a battery in the form of a big ball of starch. That big ball of starch then feeds that plant for the next year as it's growing and making new leaves and pushing itself out of the ground and running new root systems and all the stuff that a potato needs to do. And then you have glycogen, which is a similar thing, but it's in animals. And then you have cellulose, which is cellulose. It's more of like the lattice structural form of the plant. So plants don't really store energy in their cellulose. They use their cellulose to build their structure. So you don't have much reabsorption or breakdown of cellulose in a plant back into its energy form. They use starches to do that. So if you think about like us using our fats or our glycogen to store energy, plants use starch in the same way. And then cellulose is more like our bones for a plant, if that makes sense. It's their structural cells for the plant. It's the cell walls are all made up of cellulose. And cellulose is very complicated. It's this big long chain of these carbohydrates, and it's why it's really hard for animals to break it down. It's why cows have multi-chambered stomachs and have a whole process where they ruminate on food, and that food, that grass then gets broken down by all these bacteria and other organisms that live in their guts to basically go through the process of eating grass and turning that grass into a sugar that cow can then turn into proteins and glycogen and other things in its body. But it's something that we can't do. We don't have the biology in our gut or in our body to take raw cellulose and turn it into anything. And it's why, like with celery, you don't get anything, you hardly get any sort of nutrients out of eating celery. It's mostly cellulose and water. We can eat it, and we can, it's not toxic to us, but basically you don't break it down into any sort of usable energy or product. You just eat it and pass it through your body. And part of that is, everybody poops, right? That's the name of the little kid's book. But that's important. That process in mammals, and especially in omnivores and carnivores, is an important process, in that that cellulose actually kind of acts like a bit of a scrubber for pulling, for cleaning your gut out. And it's, it's why they recommend that you eat a lot of fiber in your diet. Even though you don't actually get nutrients from that fiber, that the structure of the fiber is important for people especially, but a lot of mammals for their gut health. And it's also important for the bacteria that live and thrive inside of your body. Because if your gut is just, and they find this, they're every year, it seems like they're expanding on this knowledge of like the impact that the flora of the bacteria in your gut have on all kinds of things, including how your brain works. So, so all of these things are very, they're all tied together, very important. We're just going up in complexity here. We'll see the same thing when we talk about the proteins and the lipids. I'll start with the basics, and we'll expand these out to their more complex forms. So here's some of the polysaccharides. I just talked about that. So you have this starch chain that is formed in potatoes, and you can see that it's actually quite similar in structure to the glycogen. Glycogen has it's not these single chain structures; they'll branch, which is what's different in people, and it's in your muscles, in your liver, and it's like a readily available energy source without having to break down fats into their base products to then use those fats. And then we have cellulose. And like I said, cellulose, you can see that you have these multi-chains that are all linked together. And this whole thing is a very complex structure, and it does take a very special process for this to break down. What kind of carbohydrate is our single ring sugar? Glucose.
Not monosaccharide.
Yep. And the reason behind that is that's our smallest piece, right? And then our disaccharides are 2, and polysaccharides are the chains.
Chains, yeah.
We're going to look at our glycerols. And these are basically the lipid side of this process. So I should have switched this around a little bit. But you have your glycerides are your small part of this structure, and a lipid is a triglyceride. So you have 3 glycerides make up your triglyceride, and you can see these up here at the top of the screen. I think I've got. Okay, yeah, in this structure right here, this single piece here is a glycerol. Okay, and so this, like your simple sugars, is your basic building block. And so a lipid is a triglycerol. So 3 glycerols make up a lipid. And so you remember how I said you have these zigzaggy lines? A lot of times you'll see your lipids represented without the carbons shown in here. So it will, and without the hydrogen shown in here. So you'll have this group up here at the top, and then you'll just have a zigzaggy line that goes all the way out. And so just keep in mind that anytime you have that, it's representing a carbon with 2 hydrogens poking off of it, okay? So, and if you have a double line going between your carbons, it means that you don't have 2 hydrogens, you only have a single hydrogen poking off of those. And all of that changes how those structures are bent and collapse in on each other, and how they function and how they bond with things in future use. So your saturated fats, there's a list of them up here. And one way to tell if something's a saturated versus an unsaturated fat is your saturated fats at room temperature tend to be solid. So if you think about like butter and cheese and coconut oil and things like that, that at room temperature, they're a solid. And I know they have milk on here, but it's really more of the cream layer on top of the whole milk that's a saturated fat in there. Because when you separate out the fat of milk, so like you get 2% or 1% or whatever, what they're talking about there is how much cream they're pulling out. And if you grew up around cows, which I did, if you take raw milk and you put it in a gallon jar and you set it in a refrigerator, in a gallon jar, you'll get almost probably 3/4 of an inch to an inch of just fat that comes up to the surface of that milk. And that's the cream. And that's a big part of the energy that's in that milk that is designed for a baby to grow. So, I mean, if you think about it, you know, that's the entire point of a cow producing milk, is to feed their baby for the first part of their life until they're ready to break down grass and things on their own. So, and you think about the size of a cow, there has to be a ton of energy in there for that to happen. So in a gallon of milk, there's a ton of energy, and that energy is stored in the form of a fat, and that fat is a lipid. So if that makes sense. And you know, so, and you can see that if you even if you take whole milk, which has been pasteurized, and there are a whole bunch of other things to it, if you leave it in your refrigerator and you open up the lid and you look in, you'll see a cream layer on top of that whole milk. So that cream layer will separate out and float to the top of the milk. So, and then your unsaturated fats, these are the ones that don't have that bond in them. They tend to be liquid at room temperature. And you can see that in both cases, we have some that are animal-based and some that are plant-based. Your unsaturated fats are, for the most part, except for fish, are mostly plant-based. So, all right, so let's look at proteins. So proteins are very complex. So do you remember how I was saying there's, when you start looking at structurally, how these things start to put together? This is a protein here, and they form like these ribbons, and then they have long chains, and all of them kind of stack and curl around each other and fit into these really weird structures. But in most cases, it's a very functional weird structure. Once again, it shows just how chaotic and crazy evolution is. That, you know, that this through, you know, millions and millions and millions of years of evolution is what has worked. But yeah, very, very cool, very neat stuff when you get into some of the upper level classes that talk about why this sort of stuff happens. And we'll talk about it a tiny bit, but not much. So we have amino acids, and amino acids are your base monomer for proteins, right? So you get chains of amino acids together, and those are going to be your polypeptides. And then from there, your polypeptides are going to make more and more complex systems. So, and you have, like you can see here, you have, I mean, basically everything in your body, because we don't make cellulose to create our structure. We have proteins instead. So we have this amino acid base instead of a sugar base necessarily. And then, you know, we're not made up of carbohydrates. So this amino acid base is what is the base for all of our muscles and our bones and our blood and all of these other things. So what you can see here is how so basically this is from your very basic structure, and this is like looking at the single rings for sugars and how they formed into longer and longer chain structures. This is the same idea for proteins, except for amino acids are already in chains, and then those amino acids form a secondary protein structure where you get these like helical sheets or pleated sheets, and then those form a tertiary structure where they kind of then twist in with each other and make another structure, and then you get a quaternary structure which is multiple proteins and others that kind of bond together. So it's, once again, it's from a very simple structure into these much more complex structures, though you don't have the rigidity that you have a tendency to find in, like, cellulose is a very rigid, very linear chemical structure, and you see that in plant cell walls. Plant cell walls are square, or you know, they're set at angles to each other, where, you know, animal cells and you know, other organism cells are they have a tendency to be a lot messier in what they look like. They're a lot more fluid in their structure. It's what allows us to have movement. If we were made up of cellulose, we'd have very little movement. You think about, some plants do move when they follow the sun. A lot of that has to do with water movement across the cells, and actually deflating and inflating cells to get them to collapse, to get them to move around things. And so, like, the ability to have movement isn't 100% reliant on this kind of messier structure, but it definitely makes that a lot more available. You also then run into some size limits, and that's why you can have massive giant trees, because they're a much more rigid structure. And our big giant animals, like the biggest of our giant animals, have to be supported, their structure has to be supported by water, because they can't physically support their own weight. So it's that trade-off between a rigid structure of cellular building blocks versus a fluid structure of cellular building blocks. Okay, so why would a protein be an example of a macromolecule?
Because it's a chain of nucleic acids.
Amino acids, but yes. So it's your amino acids would be your monomers, and your proteins are your macromolecules. So they're the chain together, or multi-structural pieces. So if you think about it, almost like Legos. Your monosaccharides would be those little individual singular blocks, and your macromolecules would be, you know, a more complex block. And then your animal would be the thing that you built in the end that was made out of all of those blocks. So here's our nucleic acids. So this is the other one. And these are your DNAs and your RNAs. So you have a sugar. Do you remember how I said that? And it's a 5-carbon sugar, a phosphate group, and your nitrogenous group. And these are your ATCs and Gs that when you build up your DNA chains, you see a DNA structure. They link together over on this side as the outside of that helix. And then your inner bridge, like your ladder structure that's inside, are your nitrogen-based structures touching together. And I can't remember if it's on this slide. It is a little bit. I might have a little bit of a different slide that actually shows us a tiny bit better. But you can see here off of this phosphorus structure, over here, you have the phosphorus structure is then binding to the sugar of the chain that's up above it, right? And so you get this stair-step binding along the outside, and that's represented typically in a drawing by this ribbon. And then you have your nitrogenous structure, which are the ones in the middle. Here's a 3D model of that. And you can see, once again, this outside structure is your phosphorus chains, and you get 2 of them. And then your inside ladders are the nitrogen chains, and then they're all linked together by these 5-carbon sugars that structure in the middle. And then this one kind of follows along before. Anybody got an answer for that?
Because nucleic acids make DNA and RNA.
Exactly. So your nucleic acids are your monomers, and then these are your macromolecules. So these are the molecules that are made up of your smaller building blocks. All right, so that's what I've got for that slideshow. Do you